To start, we will go to the cell cytoplasm and find ourselves some glucose to break down. Here's a cell, here's a cytoplasm, and here's some glucose. The first step in glycolysis is converting the glucose into glucose 6-phosphate. Essentially, you take hexokinase, take the phosphate group from ATP, turn it into ADP, and put the phosphate group onto the 6 carbon, thus creating glucose 6-phosphate. You get your ATP, which stands for adenosine triphosphate, and you put that into that 6 carbon. The next step is using the enzyme phosphoglucose isomerase, which basically takes glucose 6-phosphate and breaks it from a 6-membered ring into a 5-membered ring. And essentially, this enzyme breaks up carbon 1 from the oxygen group, and then the oxygen bonds with carbon 2, making you a beautiful fructose 6-phosphate molecule. The next step is taking the fructose 6-phosphate using ATP with PFA1, which stands for phosphofructokinase, a rate-limiting enzyme, turning that into ADP by putting that phosphate group onto the carbon 1, giving you fructose 1,6-bisphosphate. The next step is using the enzyme catalase, or fructose 1,6-bisphosphate catalase. There are 2 phosphate groups and the 6 carbons, and this enzyme catalase basically cuts the carbon 3 and carbon 4 apart, and so that you get dihydroxyacetone phosphate, or DHAP, and glyceraldehyde 3-phosphate, or DAP. And you can see that carbon 4, 5, 6 is together, and 1, 2, 3 are together. Then you have DHAP converting to DAP using triose phosphate isomerase. So DAP is the only molecule that can continue in the glycolytic pathway, thus all DHAP molecules turn into DAP molecules by this triose phosphate isomerase. So now we have our DAP, which is going to be transformed using glyceraldehyde 3-phosphate dehydrogenase with nicotinamide adenine dinucleotide to create nicotinamide adenine dinucleotide plus hydrogen to create 1,3-biphosphoglycerate. And you'll see that there's now 2 phosphate groups on this 3-carbon chain. Note that we have 2 DAP molecules from the 1 glucose, so we end up making 2 NADH. The 2 molecules of 1,3-biphosphoglycerate will have a reaction with the enzyme phosphoglycerate kinase, or PGK, to dephosphorylate this molecule and add it into the ADP to create ATP. And you can see that now it becomes 3-phosphoglycerate. The next step is now the 3-phosphoglycerate will go and react with phosphoglycerate mutase to change the phosphate group from the 3rd carbon into the 2nd carbon. Then using the enzyme enolase, the 2-phosphoglycerate will have the water removed from that molecule, specifically the 2nd carbon and 3rd carbon's hydrogen and respective alcohol group will be removed, and then you'll get a double bond with an oxygen on the 1st carbon and a double bond between the 2nd and 3rd carbon and the phosphate group there. This leaves us with phosphoenolpyruvate, and then the pyruvate kinase will remove the phosphate group from the 2nd carbon to create pyruvate. Like all in all, we have glucose going through glycolysis to create 2 molecules of pyruvate, which results in using 2 ATPD to produce 4 ATP and giving us a net production of 2 ATP and 2 NADH. So with 1 molecule of glucose going through glycolysis, you'll get 2 molecules of pyruvate. Now we got to figure out where do these go after glycolysis. After glycolysis, if there's no sufficient oxygen, pyruvate reacts with lactate dehydrogenase to become 2 molecules of lactate. And then you'll have NADH to become NAD+. Now, if there is sufficient oxygen, the pyruvate formed during glycolysis will enter the mitochondrial matrix and undergoes an oxidative decarboxylation to form 2 molecules of acetyl-CoA. In this reaction, the pyruvate dehydrogenase enzyme will act as a catalyst, and CO2, or carbon dioxide, and NADH are produced in this step. So again, you have 1 glucose making 2 pyruvate, and the 2 pyruvate will make 2 acetyl-CoA. So in the Krebs cycle is where your acetyl-CoA will go, and acetyl-CoA will react with oxaloacetate to make citrate, and it'll use the enzyme citrate synthase. And you can see that the oxaloacetate plus the acetyl-CoA will give you 6 carbons. So citrate will have a hydration reaction with aconitase to make the isomer of citrate, which is isocitrate. And then from there, isocitrate dehydrogenase, which is the rate-limiting enzyme for the Krebs cycle, will react with isocitrate, producing CO2 and NADH, and it will make alpha ketoglutarate. And you can see that there are fewer carbons on the isocitrate because of the carbon dioxide removal. So we have alpha ketoglutarate dehydrogenase reacting with alpha ketoglutarate, making NADH and CO2. And there's going to be an additional coenzyme A to make succinyl coenzyme A. And you can see that there are now 4 carbons and a coenzyme A. Then succinyl CoA synthetase will take GTP and an inorganic phosphate to make GTP. And GTP will inadvertently turn into ATP. Succinate, which has its coenzyme A already removed from the previous reaction, will react with succinic dehydrogenase to turn FAD, which is flavin adenine dinucleotide, to FADH2, which gives you fumarate. And then fumarate will react with fumarase to make malate. Malate will react with malate dehydrogenase to turn NAD plus to NADH to get back to oxaloacetate. And there's your Krebs cycle. In summary, the Krebs cycle, otherwise known as the citric acid cycle, produces 1 GTP, which converts to 1 ATP, 2 NADH, 1 FADH2 per turn. Thus, from 1 glucose molecule, we get 2 pyruvate, translating to 2 acetyl-CoA, and therefore 2 cycles within the Krebs cycle, resulting in 2 GTP, 6 NADH, 2 FADH2 per glucose molecule. So in total, we'll have 4 ATP, 10 NADH, and 2 FADH2. So where does our energy currency, aka ATP, and electron carriers, aka NADH and FADH2, go? Electron transport chain. And as you can see here, you see the cytoplasm, the outer membrane, the intramembrane space, the inner membrane, and the mitochondrial matrix. This is the electron transport chain. NADH gets reduced to NAD+, FADH2 gets reduced to FAD+. NADH will donate its high energy electrons to complex 1, which promotes the pumping of a hydrogen through complex 1. And as the electrons travel down, FADH2 will donate its lower energy electrons to complex 2. And as it travels down complex 3, hydrogens get pumped through that. Cytochrome C will have the electron travel through all the way to cytochrome C oxidase, which is the rate-limiting enzyme, and that will also pump hydrogens. As the electrons move downhill through the ETC, complexes 1, 3, and 4 will pump protons from the matrix into the intramembrane space, creating an electrochemical proton gradient. The proton gradient then drives ATP synthase, which is a rotary enzyme that allows protons to flow back into the matrix. This proton flow powers conversion of ADP plus an inorganic phosphate into ATP, producing the majority of cellular ATP. So at complex 4, cytochrome C oxidase, the rate-limiting enzyme of the electron transport chain, electrons are transferred to the molecular oxygen, which is the final electron acceptor. Oxygen combines with electrons and protons to form water, preventing electron backup in the chain and allows for continued respiration.